As we wait for more attendees to join our webinar, I'd like to take this moment to explain about our ACRAS project. Okay. So let me start again. My name is Yuta Sekino. I'm with National Cancer Center Hospital Japan and Asian Partnership Office, so-called NCC Japan Bangkok Office. So let me start with my brief introduction about our project. Our project, ACRAS, stands for Asian Clinical Trials Network for Cancers. And our main objective is to establish a robust platform for international clinical trials in Asia. In September 2020, the National Cancer Center Japan initiated this project based on the existing network in Asia with support from the Japanese government funding agency. And so far, we have fostered partnerships across 10+1 countries and regions, mainly ASEAN region, and one from European organization, Europe, and involving 14 institutions, and each are leading cancer treatment facilities from distinct geographic areas. And we have more than 420 active members, including investigators and study coordinators in the network. And currently, five conducting ongoing clinical trials, and more clinical trials are under discussion and planning with the pharmaceutical companies. And we have enrolled more than 2,700 and more patients enrolled. And to strengthen ACRAS as a shared platform, we have established the ACRAS board. This board governs all strategic decision and operations. Currently, ACRAS board is the president of ACRAS board is Dr. Darren Lim from Singapore, and president-elect is Dr. Tom Wei-Wun Chen from Taiwan, and also quite famous investigators in SARCOMA field. And also, we have to accelerate treatment development for disease specific to Asia. We have launched the ACRAS study group. Currently, we have 3 groups and 36 members. And the first group was head and neck cancer group, and the second one is sarcoma rare cancer group. And Dr. Barry is a member of the sarcoma rare cancer group, and she's one of the most active investigators in our group. And in addition to the website, we regularly update our Facebook and X, former Twitter, and LinkedIn pages with information about the webinar and also our progress. So please follow us, and I will post the links in chat shortly. And before we start the main presentation, a few housekeeping notes. The first one, we warmly welcome questions and comments. Please use either Q&A functions or right-hand feature under the reaction. And also, we are to make our webinar more interactive. We encourage attendees to use reactions throughout the webinar. Thank you so much. So let's practice now, and please send any reaction you like. Thank you so much for your cooperation. Okay, so that concludes the introduction of the ACRAS project. And I'm now pleased and honored to introduce our moderator for today's session, Dr. Barry Yan. She's belonged to, and as I mentioned, one of the activists that most active investigator in sarcoma rare cancer group, and from OncoCare Cancer Center, Singapore. Now, the floor is yours, Barry.
Thank you so much, Yuta, Dr. Sekino. Good evening, everyone, and actually good afternoon and good morning to those of you from different parts of the world. I think there are even attendees from the US and Italy who are on the call as well. So thank you very much for joining us today for what is going to be a very exciting topic in this very somewhat neglected and little known group of cancers, right? Sarcomas and rare cancers. So this is really our specialty interest, and it's my absolute pleasure and honor to introduce our very first speaker. So Dr. Wong Rusin is a radiation oncologist, actually, and she is now at the Icon Cancer Center in Singapore. She remains a visiting consultant at Proton Therapy Singapore. She graduated from the National University of Singapore and trained in radiation oncology at the National Cancer Centre Singapore before joining private practice. She actually is a wonderful colleague of mine for many years. She's held multiple grants in sarcomas. She's worked on multiple pediatric projects, and she has a keen interest in precision radiotherapy for sarcomas and these other rare cancers. So very apt for her to speak on today's first lecture, which is that of precision radiotherapy for sarcomas and other rare cancers. Dr. Wong, please.
Thank you, Dr. Valerie. Let me share my screen. Okay. All right. Is it shown?
Yes, we can see it. Thanks, Rusin.
Okay. Hi, everyone. So the theme for today's webinar is sarcoma. So I'll be talking about precision radiotherapy, specifically with proton beam therapy, with stereotactic body radiotherapy, and how to sequence these treatments with biologics. So I was told to do a 5-minute introduction of my facility, followed by a 15-minute radiotherapy talk. I have no disclosures. So I work in Mount Alvernia Hospital, Singapore. Our facility wise, we have a variant true beam radiotherapy machine with hyper-arc software capability and identify, which is for surface guidance. I know a lot of technical terms. We also use artificial intelligence for auto contouring to make my life easier. And because Icon Cancer Center is an international group, so we also have to follow guidelines. We have an in-house Siemens PET-CT with respiratory gating for tumors that move. And I also treat patients at Biopolis, Proton Therapy Singapore. And in that center, I use a variant probeam machine with an in-house PET-CT and MRI. So before I move on, let's talk about stereotactic body radiotherapy. So in a very simple concept, this is an ablative treatment delivered with surgical precision, but without a knife. Okay, and just a very quick introduction to proton beam therapy. It's a form of external beam radiotherapy, but using protons. Right, sorry, I have incoming call. Cancel it. Okay, and using protons, so the protons create what we call a Bragg peak, so that the energy is deposited at a controllable depth, meaning the tumor, and there's minimal exit dose beyond the target. This is good for sarcomas in certain locations because we can dose escalate to give a higher radiotherapy dose to the sarcomas, which can be radio resistant, and also to avoid sensitive organs. So for stereotactic body radiotherapy, I use surface guidance. Nowadays when you go through gates, gantries, you have facial recognition, and likewise we have body recognition to get the patient's body shape so that we can deliver precise radiotherapy. And for organs that move, the liver, the pancreas, the lungs, we also have breath holding software. For example, over here, the patient sees a screen and the patient is told to hold his or her breath. And likewise, in the proton therapy center, our machine is 360 degrees, so it goes around the patient. This couch is a robotic one. There are 6 degrees of freedom, so it can move in 6 angles. We also have CT that's mounted onto the gantry so that we can take images during the treatment for precision. And there's also motion control as well for tumors that move. So in Singapore, approved sarcoma indications for proton beam therapy with subsidy for all ages would be sarcomas in the base of skull, in the spinal or paraspinal region, retroperitoneal sarcomas, and also for locally advanced sarcomas which require concurrent chemotherapy. And for younger patients with Ewing's, with rhabdomyosarcoma, osteosarcomas, and also pelvic sarcomas. So these are all eligible for subsidies. So I will go through a few patient anecdotes. This is a patient of mine, a teenager with chest wall Ewing's over here. So she also had malignant ipsilateral pleural effusion. She had dose compressed chemo, followed by resection, and then she had proton beam therapy, not only to the primary, but also to the entire hemithorax. And the point of using proton beam therapy is so that we can reduce dose to her heart and also her breast, since she's a young teenager. Okay, and this is a patient of mine with Dr. Valerie, young man with Ewing sarcoma of the spine with metastases to the entire spinal column and also to the pelvis. So with proton beam therapy, I was able to treat not only the primary, but also the entire vertebral column and spare the anterior thoracic, abdominal, and pelvic organs. This is a patient that belongs to my colleague who also had a spinal E-wings, and this patient had surgical instruments inserted in. So these surgical instruments actually can interfere with proton beam therapy. So the physicist had to do commissioning works to make sure that this are accounted for. This is the patient of mine with a very big sacral chordoma, and with proton beam therapy, we could avoid his anterior pelvic organs. This is a lady who had a sacrectomy, but after that relapsed locally, and the surgeon put in a omentum spacer. You can see over here the fats so that her rectum and her anterior organs are pushed away from the radiotherapy area. And with proton beam therapy, we could then avoid all these organs anteriorly. Another gentleman with base of skull chondrosarcoma, and this region is very tricky. You can see all the cranial nerves coming out actually, and with proton beam therapy, I could not avoid the cranial nerves, but the goal was to avoid irradiating the brainstem, which is a radiosensitive organ. And also, chondrosarcoma requires dose escalation, and we could do that with proton beam therapy. This is another challenging case, 60-year-old gentleman that I co-managed with my colleague. He had a large, unresectable, undifferentiated pleomorphic sarcoma of the pericardium that was cystic. So he had chemo followed by proton beam therapy. And because this region moves with respiration, so we had to take that into account. And the cyst was changing size during the 7 weeks of radiotherapy. So we also had to do replanning to make sure that the treatment was accurate. And I was told that 3 years on, this patient is still disease-free. Okay, other than primary sarcomas, I will also like to talk about radiation-induced sarcomas, which have received radiotherapy before, as well as local relapses of sarcomas, which also have received radiotherapy before. Okay, this is an elderly male who had nasopharyngeal carcinoma, which is a cancer that is endemic in this region. And more than 10 years later, he unfortunately presented with proptosis. His eye was bulging out and there was a sarcoma growing in the maxilla. So this was most likely radiation-induced sarcoma after more than 10 years of chemoradiotherapy. And he didn't have many options. Surgery was out of the question. So I opted to give him re-irradiation. So he survived with some toxicities, mainly cranial nerve neuropathies, as well as inflammation and infection of the base of skull, which is managed with long-term antibiotics. Okay, this is a paper that I wrote with Dr. Valerie, an elderly lady with a chondrosarcoma of the shoulder, which had radiotherapy before. She relapsed, and then I gave her what we call pulse low dose rate. The concept is that with pulses, rather than just one shot of radiotherapy, each time we will give 10 times to lower the dose to protect the normal tissues. And this is another elderly gentleman who had radiotherapy for pelvic sarcoma. He relapsed, and then when we relapsed, I gave him pulse radiotherapy. And this gentleman is still alive and well. Okay, so moving on from proton beam therapy, I'd like to talk about stereotactic body radiotherapy. So I mentioned before, what it is it's like GPS with positioning, with imaging. We want to make sure that the position is accurate because we give very, very high doses of ablative treatment. In sarcomas, this is indicated when there's oligoprogression, meaning in a metastatic sarcoma patient, only a few metastases are progressing, or in patients with oligometastasis, meaning a limited number of extra primary sites of spread. So in these patients, we can still give very high dose ablative radiotherapy to try to cure and control these areas of metastasis. And increasingly, there are some case series, some larger than others, to show that actually for sarcomas, this can give survival benefit. So to recap, today's theme is precision radiotherapy for sarcomas. So the tools and the toys that we have are artificial intelligence planning, image guidance with in-house PET-CT, MRI, and also we have CT mounted onto the machines. And for organs that move, we also have breath and motion control. And with precision techniques like SBRT and proton beam therapy, we can give our patients more precise treatment. Okay, so precision treatment needs to go hand in hand with targeted systemics. So we all know that immunotherapy and biologics are a rapidly evolving field, and many times I have to think about how to sequence radiotherapy with these biologics because there's increased risk of toxicity due to biological interactions. I'd like to bring your attention to this framework published by the ISMO-ISTRO group 1 or 2 years ago. And essentially, there are 4 classes of biologics. You have biologics that interfere with DNA repair, and this can actually cause increased toxicities. You also have biologics that are proliferation inhibitors, and for fast proliferating tissues, this can actually cause increased toxicities. For immune checkpoint inhibitors, we are actually quite confident that radiotherapy can be given concurrently, but there's a slight potential increased risk of late inflammation. With angiogenesis inhibitors, it depends on where we're giving the radiotherapy. If it's to musculoskeletal soft tissue, we know that the toxicity is slightly increased and probably manageable. But for treatments near tissues like the brain, the gastrointestinal, the urinary tract or respiratory tract, we are a little bit more wary because this can cause perforation and even lethal bleeding and fistulas. So I use Google AI to come up with this infographic. Essentially, there's DNA response amplification interaction with radiotherapy and biologics. There's also normal tissue repair inhibition. And if we do need to stop biologics, we can consider the 5 half-life rule, which is to wash out the biologics before we give our radiotherapy. But I think overall, we have to balance risk versus efficacy in these patients. So we always need clinical judgment. And this paper just came out a few days ago, actually. This is real-world data. So this is a European prospective multi-center registry with 433 patients, and they all had metastasis-directed stereotactic radiotherapy, and they also had concurrent biologics. Some of them had immune checkpoint inhibitors, some of them had small molecules, some of them had monoclonal antibodies. And about half were cranial and half were extracranial. And after 2 years of follow-up, they found that actually grade 3 toxicities were less than 10%, but 1.2% of patients actually died from bleeding, and these were all in cranial stereotactic radiotherapy patients. So if you look at both the ISTRO and ISMO framework, you can see that there's tie-in with real-world data to say that actually, BRAF inhibitors can slightly cause risk of bleeding, grade 5 bleeding in cranial SRS. This is most probably in melanoma patients, maybe not so common in Asia. And also for patients with EGFR inhibitors, there's a signal that there's a late toxicity increase. Okay, but having said that, it's still less than 10%. So because this is an Atlas webinar, I'd like to take this opportunity to use the platform to say that going forward, our aspiration is to foster sarcoma programs. So we need collaboration. Thanks, Yuta and team. We need networking so that we can pool expertise and resources for these rare tumors. And networking can also clinically benefit patients if we can have seamless referrals, clinical trials access, and this is a very good global health initiative so that we can all access knowledge and resources. And proton beam therapy is increasingly more and more available in some countries, but there are also many countries in Asia that do not have access. So if we can establish referral systems to coordinate patient care, this would be very ideal. And we also have Asian groups like FARO, PT-COG, and Atlas to formulate, hopefully, collaborative proton beam referral programs and research and logistical programs. And as well as there's also the Southeast Asian Radiation Oncology Group. Okay, so that's my time. Over to Valerie.
Thank you very much, Rusin. That was a fantastic tour de force, actually, about precision radiotherapy for sarcomas and rare cancers. Thank you for simplifying such a complex topic into something that is so digestible. I think the cases that you listed as examples are really stark, actually. I think maybe for those of us who are not so used to seeing what radiation planning is like, it's really not easy. So thank you very much for the wonderful talk. I think we have time to possibly take one question if there's any burning questions right now. If not, we will reserve questions for later. I have many questions for Rusin, actually. Maybe not. Okay, actually, Yuta, do you know how do we tell if people need to ask questions? I'm not sure.
Can you see the Q&A function on your toolbar?
Ah, yes. Okay, my bad. Okay, I see it. Okay, so I think there's a comment. Yep. Okay. I think there was a clinical question about radiating lesions in the brain. And then, okay, so maybe shall we take the one question for Rusin first? Rusin, there's a question about, is there a standardized consultation or referral system for proton beam therapy in Singapore?
Okay, standardized. There are 3 centers in Singapore, so I guess there's no one referral to all 3, but I guess it will be quite easy to reach out to me. I can't speak for the rest. And we have clinical coordinators to help with case notes collection, DICOM retrieval. We can also do teleconsultations before patients physically arrive. I hope this answers the question.
Yep. Okay. Thank you, Rusin. And I think certainly there is also there are multiple proton beam tumor boards as well. So all referrals that come in will be cleared through a tumor board so that it's approved. Am I right to say?
Yes, and Valerie on there are a few of them.
Yes. And increasingly, there's also expanding indications for the use of proton beam in Singapore, right?
Yeah.
Okay, wonderful. Thank you. There's one other question, but maybe we'll reserve that for later if that's okay. Yep, okay. The interest of time, I will go back to that later. Okay, so thank you. It's my pleasure to introduce the next speaker, wonderful colleague of mine who have been working together for many years, Dr. Wu Xingyi. Dr. Wu is a senior principal investigator at the Bioinformatics Institute, part of Singapore's Agency for Science, Technology and Research. She leads research data integration and platform development initiatives across translational and clinical research across multiple disease areas. She completed her doctoral training at University of Illinois at Urbana-Champaign and National University of Singapore. She specializes in computational modeling and systems engineering. So she then transitioned into cancer genomics during her postdoctoral research at the Genome Institute of Singapore, where she analyzed large-scale sequencing data across diverse tumor types under the mentorship of Professor Edison Liu and Guillaume Bourg. Her work contributed to key discoveries in cancer genome architecture, including the identification of a distinct tandem duplicator phenotype and mechanisms underlying amplicon formation in breast cancer. Xingyi then spent nearly a decade at the Jackson Laboratory in the United States, where she played a leading role in developing large-scale cancer genomic workflows, knowledge bases, data commons. And she was a core contributor to the NCI PDX-NET Data Commons and Coordinating Center, and supporting multi-institutional data integration efforts across North America, Europe, and Asia. So in 2022, where we then first worked together with Xingyi, was when she established her independent research group at the BII, and where she worked closely with clinicians like myself and experimental scientists like myself too, to develop scalable data science platforms and translate multimodal data into clinical impact across cancer, actually also across ophthalmology, dermatology, metabolic and mental health disease, as well as biopharmaceutical manufacturing. So her work has generated publications in multiple journals, including Nature Genetics, Cancer Research, Genome Research. So it's my pleasure and honor to introduce Singi, and her topic today is really quite a big topic and very important as well, to look at how we can push this to patients for these sarcomas and rare cancers that sometimes have very limited treatment options. Data integration, multi-institutional efforts for translational sarcoma research. Thank you, Singi.
Thank you, Valerie. Can you hear me? Okay.
Yes, we
Can
can.
you see my slides?
Yes.
Okay. Hi, everyone. Thank you, Atlas and Dr. Yuta, for the invitation to talk about our work. So let me just give a brief introduction on the Bioinformatics Institute, where I come from. So this is part of a larger A-star government-funded institution, and we are housed in this matrix building in Biopolis. So currently, Bioinformatics Institute, or BII for short, has 19 research groups, and we are fully computational, and we house close to 130 experts in various expertise of bioinformatics, computational biology, AI, and data science. And we are also still actively expanding and recruiting. So Dr. Sebastian Morris-Stroh is our executive director. As you can see, we have a very ambitious vision to really lead the AI and data science and analytical efforts in Singapore biomedical and biotech ecosystem. So what we want to do as a mission is really to look at biological and healthcare data and creating value from them. So we have very strong international connections as well. We have various key programs now, working on precision health, drug discovery, cellular biotechnology, synthetic biology. We are also running a data hub on biomedical data, and we are actively engaging in new AI emerging technologies. And we are also in the pandemic preparedness program. So in terms of international connections, the few that we have is working with HDR UK, and we belong to consortiums that is actually very active in terms of data sharing across multiple countries, including like Taiwan, Japan, and so on. And of course, we are actually one of the data centers for GSAID. If you remember from COVID, it's actually all the data actually flows through BII to inform World Health Organization on emergence of new variants. And just a little one slide to let you know what I actually do in BII is that my group is research data integration. So we really work with large collaborative programs to coordinate their data, analyze their data to help them drive translational impact. So if you can see, all of us are being working with clinicians and all that, we know that every single patient with the disease can generate a lot of data, both all from the clinical records to images in the hospitals, as well as additional surveys and all that. And from there, we can also, retrieve lots of different samples like blood samples, tissue samples, and all that. And if we work with a wet lab, all these go into building patient-derived models, and this can be patient-derived cell lines, PDX in the mouse, or zebrafish, and so on. So this gives us a whole extensive cellular libraries for us to do a lot of different molecular screens. From patients, we also could have clinical trial data, and all these different screens and samples, whether from the patient models or different cell lines, with all the advancements of multi-omics technology, we can put it through different types of profiling. So you can see that, from one single patient, the complex of data coming from one single patient of a particular disease. And when we scale this up to a very heterogeneous patient group, especially in sarcomas of different subtypes, different demographics, and we can also collect data that is, of longitudinal data. So becomes, a large complex data set that any, computational group that has to deal with. So the goal of my group is really to, play the role of being a coordination center with these large programs to carry out, end-to-end research data integration. So other than working with our IT teams on a lot of data security, we really take on many tasks from managing the data, analyzing, integrating them, as well as working with our partners, our collaborators into interpreting and visualizing the data. So if data comes in well organized, that's where we can pursue, AI and international collaborations. So in Singapore, we are also, we have many platforms, including our Ministry of Health. There's a platform called Trust on government data, as well as a Singapore Translational Cancer Consortium, where we link our projects together with them. And as I was mentioning earlier, the HDR UK is part of our HealthWise consortium that we connect with many countries. And we know that many countries like to store their data in these trusted research environments. So we are also exploring, how all these data could be connected between different countries as well. So with this whole framework, I work on a number of projects, including IO-treated pan cancer, colorectal cancers, lymphoma, and so on. So sarcomas with Dr. Valerie is what I'm going to talk about today, as well as the Genomics Center is on large population sequencing. But I also dabble on other diseases like atopic dermatitis, age-related macular degeneration, fatty liver disease, obesity, and so on. And we also extend a lot of these to biologics manufacturing that actually generate a lot of data that's related to manufacturing bispecifics for cancer treatment as well. So as you can see, this is really a multi-institutional work throughout Singapore. So I'm sure all of us here are experts in sarcoma, and we know that it's well known to be called the loneliest cancer because it only affects less than 1% of adult cancers and more in childhood cancers. And it's difficult to study because given such a relatively small number compared to different cancer types, there are actually more than 50 distinct subtypes. And it's very underrepresented in research studies, extremely heterogeneous in terms of molecular profiles. And the key need on why we really need to push this is because when it comes to especially metastatic disease, the dismal response rate of less than 10% is happening to our patients. So that's why we need really a multidisciplinary team to bring together different experts and to really explore how precision medicine can help our patients. So sarcoma is the loneliest cancer, but I have a very big group of researchers to work with. This is our current group that we work with now in Singapore. This is really multi-institutional, as you can see, and multidisciplinary. We have a strong clinical team from medical oncology, the surgeons, radiation oncology, as you listened to Rusin just now, as well as we work actively with pathologists and epidemiologists. And so my team here is the really data integration team, but we also connect actively with the universities as well as other A-star institutes on building models, molecular biology, immunology, and so on. And so as you can see, this is also a very young team. And what you're going to see in the next slides coming up is really a lot of work starting, after COVID. With some of us starting this work, and we are coming together just in the past, 2 to 3 years to really drive this research. So with the data coming in and all that, I was able to see through a lot of meetings and conversations with all the collaborators, as you see in the previous slide, that we really divide ourselves into key themes on how we can drive this rare cancer research. So we work on the clinical epidemiologically driven by our clinicians in different sarcoma subtypes. We look at these various topics from head and neck sarcomas as well as mental health and all that. We also work on target discovery to look at drug screens, biomarker discovery, and looking for new targets. And we work on the various immune tumor microenvironment question as well as immunology to look at neutrophils, tertiary lymphoid structures, and so on. So we are also generating a lot of single cell spatial data. And we also have experts in building models in terms of both sarcoma cell lines, patient-derived xenografts, and so on. And with this, we actually do a lot of characterization and generate a lot of data. And we have groups looking at specific drug resistance, building resistant models, and so on, as well as proteomics expert looking at different aspects of proteomics profiling. So with all this, I try to really help to centralize all the effort and build a rare cancer atlas. So like a transition from the earlier slide that you see, in our perspective, what we get is, the patient here, really what we can get is different tumor samples that could have been from different time points and blood samples. And we have collect their clinical data. And from here, we can, other than profiling the actual tumor, there's possibility to look at xenograft models, cell lines, liquid biopsies, as well as commercial cell lines. From all these then extends to a lot of different profiling data as well as public data. So to manage all these, we then need to build a lot of systematic workflows, thinking of how to integrate the analysis, how to build the database, and so on. But all in all, our goal is to look at precision medicine and patient stratification. And in terms of the SARCOMA Atlas that we're building in Singapore, we have been working towards more than 10 years of data collection. So Dr. Valerie is the main driver of this with many clinicians as well. So we really go to the NCCS and really, recurate a lot of data with her team, looking at the past 10 years of data. And not only the data that we have gathered, we are also looking at, tissue resections and blood samples. And from these, we get all the different samples that is being profiled in using different technologies. And on the pathology side, we can also retrieve archival tissues on FFPE that helps us do a lot more studies in terms of like TMA, spatial profiling, and so on. So what I want to show you is the right is, all the data that we have collected so far using different omics and the number of samples that we have. And of course, with that, we also augment our analysis with a lot of publicly available omics data. So an overview for our current Singapore sarcoma cohort. In terms of clinical histories, we have more than 1,000 patients, over 50 histo subtypes. It's quite balanced between male and female. And we have different ethnic groups being represented. So I could say that, we are quite well represented as an Asian specific sarcoma cohort. And we are really comprehensive diagnosis and treatment data, including a lot of follow-up data so that we can do recurrence and survival analysis. So what you see on the right are the pie charts showing the different breakdown of the different subtypes. And so, really with all the data coming in, what I really need to work with my team is, how do I build a framework, a model, and an infrastructure system to actually assemble all these data to make sure, it's not just dumping files in a large computer, but into pointing, where the data is. Can I find the data? I want to make it usable for analysis and AI. So which data points back to which patient? So this is really about linking clinical information to the sample descriptors, because I was showing you we have lots of different types of samples, to the different omics that's being used, as to the detail on, what omics, what companies, what profiling technology do we use, and to the data itself, what files do we get, where do we store the data, how do we find them? And so what I want to show here is something, really a high level view of the data itself. And I won't bring you into the individual points, but here is our current data set. And every single column here represents a specific patient. So with every patient, I can actually collect a few demographics and clinical information. We actually have hundreds of fields, but we are showing the important ones here. And we are also able to see for every single patient, the types of omics that have been generated with all my different collaborators. And with this, we have the different sarcoma types. And as I said, we have different sample types, whether they are the frozen tumor, whether they are FFPE, whether they're normal tissue or blood. This is also shown by the different colors of these dots here. So with this, at a high level view, I can go back to all my collaborators and say, look, this is what we have gathered as a program, and how do we go forward to answer key research questions? So, so one thing that I built with the collaborators, with my team is, how do we navigate, our sarcoma omics availability that, in real time? So we have built, dashboards to share with them and update them frequently. So as you see right here, this is a dashboard that we built. And we can have various key distributions of diagnosis, some demographics, tumor of origin, what samples do we have, what files do we have, and so on. So, for example, the way to use it is, I can click on a specific subtype and look at, different samples and files generated. And I can also look at all the different outcomes, the data. So I can also look into a few subtypes and also select specific data and also screen through what are the patients and the IDs that's generated to this data. So this dashboard also allow me to search for specific patients, like the one I'm showing here. So we are looking at this patient of 006, and we are able to see, this patient, I've collected data on the different various assays and what are the files that we have. So really, this gives us a really high level view on what's being generated. But I've team members who are really looking into more in-depth visualization. So I was showing the complexity of the data, and everything like this could be actually, generated through actually a network as well. How we can see from a patient tumor what is being generated in terms of the different data types. So I'm just showing very fast video on a lot of exploration of visualization, but here we are really ourselves to filter things real time as data comes in, and we're able to see lots of distributions in a lot of different clinical parameters that we're going to see. So and then we can update ourselves with all the different survival plots as well over the number of years of data that we've collected. So what's interesting here at the end that I'm showing here is that we can actually pick each patient and show the timeline that the clinical data is given to us. Where is the time they get treatment? Where's the time they have surgery? When's the time they have different outcomes? Actually, this creates a very important linkage to our omics data, because we then need to see, when is our tumor sample being collected and whether it's pre-treatment and so on. So let me just change gears a bit and show you, the really downstream analysis that we carry out with the data that we get on top of all the organization. So this is published in the ESMO Real World Data Journal. It's really looking at a very rare Asian-centric head and neck sarcoma that we've collected data from 1985 to 2023 with just 275 patients. So it's a very well-curated data sets. So this is led by Dr. Mark with Dr. Valerie as the clinicians, as well as Dr. Janice Goh from my team carrying out the analysis. So we have collected data, really extensive clinical data of various disease extent, and you can see we can actually do all the survival plots. But here, on top of that, we actually do a parametric time to event modeling to actually develop a prognostic calculator. So as we know, there's a lot of prognostic calculators out there, but we don't have one that's very specific to head and neck cancer. So what we want to do is, create this prognostic calculator to, help, clinicians to think about their treatment options with this Asian cancer set. So so we have a little shiny app on this. You can go in and really play around with the different parameters, like whether it's radiotherapy induced and all that, and select all these different parameters and see how the median survival actually change. So this is one aspect on really clinical data analysis. We also look into functional precision medicine with the whole team. So this is work really driven by Dr. Sharon Chan. She's a PhD student with the group, and this collaboration with Dr. To Tan Boon, Dr. Valerie, and Dr. Edward Chow. And my team helped with some analysis and also data organization. So what they have is, single drug screening of these soft tissue sarcoma patient lines. In fact, our clinician use this data real time to inform treatment options for the patient. But there's also, with all these, Dr. Edward Chow is the driver of this QPOP platform where they can actually optimize and do optimized combination drug screens on the two drugs. So with this, they find that the best and the strongest efficacy is actually pazopanib and a BET-I inhibitor. So while BET-I inhibitor is not an approved drug, we go on to actually do a lot of mechanistic analysis. So what we did is actually to do drug treatment on the cell lines before and after the treatment on both single drugs. So ACD is the BET-I inhibitor combined on single drug pazopanib and the untreated cells. As we put this on a PCA plot, the untreated and the single drugs and the combination therapy actually shows very different expression profiles, indicating there's actually real biological differences from the response and driving the response. So we put this on a pathway analysis. We can also see very different pathways lighting up. And what's exciting for us is what we see, this combination actually exhibits a pro-apoptotic effects by suppressing oncogenic MYC pathways. And you can go to the paper and look at, we have done some experimental validations on the MYC pathway as well. So back to the fact that we are also building a patient-derived sarcoma resource. Up to now, we have close to 50 patient-derived sarcoma cell lines being established. So this is what really driven experimentally by Nadia and Dr. To Tan Boon. Nadia is the PhD student and Dr. Valerie. Upon receiving patient tumors, and they have actually optimized the media to actually culture these cell lines at very high success rate of 60 to 80%. So what we have now, I'm showing here, which is a zoom in of the big figure just now. For every single patient here, they have a patient-derived sarcoma cell line being developed, and we can actually explore all the different grades, the different subtypes, whether this derived cell line, is it treated before the resection or after the resection, because this affects a lot of our screening studies. We are able to break down whether they are age, sex, and so on. But we are also actively trying to characterize these cell lines. So we are looking at, whether we have whole exome, whole genome sequencing, single-cell RNA-seq, and all that at drug screens, both on the patient tumors and on the patient-derived cell lines. So this is a huge resource that we are trying to build, but we always get the question on, what is our sequencing data telling us? Is our cell lines, confirmed that, it's actually like our patient tumors. So we are very excited about this. We are starting to analyze the data right now. But in terms of mutations, a few of our patients here, these are all liposarcomas. They actually show very good concordance between the tumor and the PDSC, or the patient-derived sarcoma cells, on both passage 1 here. And even for this 81 patient, we actually profile up to patient passage 6, as well as this patient 92. And in terms of copy number profiles, these two patients actually show very good agreement, especially retaining the MDM2 amplifications. So we all can conclude at this with this data now that the PDSC are genetically similar to the original patient tumor. And of course, we went a bit further and see what cancer features are we really culturing. So we actually did single-cell sequencing of our patient tumor, the P0 passage and the P4 passage. So we actually assembled this, some of our single-cell sequencing of our 3 liposarcoma patients with public data sets. And we find that the cancer region here, right here, shown here, is actually enriched in the ASPC transcriptomic signature, which is not surprising because these are liposarcomas, but also because of the mesenchymal origin of sarcomas. And at the same time, we confirm them as cancer cells also because they are overexpressing MDM2 and CDK4, which is well known for liposarcomas. But what we are happy to see is our passage lines are actually in the tumor region. So indeed, we are culturing the tumor cells. And with at the single cell level now, we can actually estimate the number of cells that has MDM2 amplification. So this is the percentage that I'm showing on the right. But at the same time, we use this PDSC cells and we actually could validate these numbers with the fish experiments. So this shows that, MDM2 amplification actually, is very clonal nature. And from our single cell data, we are able to validate this in our PDSC lines. So, so this is a whirlwind of data talk, but what I wish to convey is that all the efforts here is really multidisciplinary, especially if you want to build an Asian and multi-ethnic sarcoma cohort, and we really need to look at end-to-end integration of the data to capture its translational value. And the well-curated data source is what we want to aim to do to explore precision medicine approaches. So with that, I thank the various institutions that I work with and funders and my group members, past and present, who have been working on this data. And last but not least, really the patients who have provided tumor samples and data. And we are a very exciting group that we actually journey with these patients, knowing out their outcomes and so on. So with that, thank you very much.
Fantastic. Thank you so much, Dr. Wu. That was a fantastic talk through, again, a lot of the work that we've done collaboratively over the years. Not just us, but many, many individuals and various members of the team across the whole of Singapore and even internationally. So with that, I think we already have 2 questions in the Q&A box, but feel free to keep them coming as we address these questions together. So I think the questions for Dr. Wong, thank you. Rusin has also addressed them separately, but feel free to ask more questions for Dr. Wong as well. Okay, so let's start with the first question from someone who's anonymous. Regarding the budget, how is the funding structured for these domestic multi-center large-scale projects? Could you elaborate on the balance between government grants and private sector contributions? Singi.
So all these grants, all these projects are really working on government-funded initiative. Singapore government has over the years been emphasizing, data a lot more. And so as I joined a couple of years ago, I think the timing is just striking right, where I can engage a lot of clinicians and say, look, I can help you bring together all this data to make it more meaningful. And private sector contributions, I would say that we are more actively working, say, for example, with AWS and all that, to look at various dashboard technologies, data sharing technologies, and so on. So like tomorrow, I'm really bringing my team for an AWS immersion session to really explore some of their new tools. So they are also very active in that field. So we are also searching for various cloud platforms and all that. And we're also happy to say explore certain platform dashboard tools. But at this point, a lot of things is coded in-house with standard programs. Yeah. Yeah.
Okay, thanks, Singi. If I may chime in also, I think specifically regarding the sarcoma work and the rare cancer work, it was really predominantly this was government funded, but there was also a not insignificant proportion of industry involvement where we collaborated with a lot of these technologies that we started using at the time were quite new as well. So a lot of them were technology awards that were given by some of the industries to use these technologies in sarcomas and rare cancer. But at the same time, there was also some philanthropic funding that really came from, patients or people who knew sarcoma patients who felt very strongly about supporting this area of cancer, which is typically very underfunded. So I think it's been a bit of a mix, would you say, Singi, in terms of our funding as well, specifically for sarcomas and rare cancer? Yeah.
Mm.
Okay. Great.
We
Okay.
are looking forward with this work to actually have more data-centered grants coming out actually. So
Mm.
hopefully that would actually convince a lot more of our funders as well as international funding agencies as well.
Yeah. So times are changing as well as to, what tends to be funded. So I think that's also very important to address as we develop. Okay, so then the next question is the data from the dashboard also used for clinical trial screening during MDT meetings? I suppose I should answer that question, right, Singi? Okay.
That was our big goal and ambition.
Yes. So in fact, thank you for asking the question, whoever is the anonymous attendee. This was the initial sort of goal, to build this as a whole. And this was the whole idea for integrating it so that, when we come for our weekly, sarcoma, multidisciplinary tumor boards, we can quickly go into the patient and say, okay, this particular patient, I have all these sets of data available, I have single cell data available, I have spatial data available, any genomics, which might be important for, actionable alterations which we can target. So this was the intention at the start. I don't think it has reached that stage at the moment where we can actually do it live. However, I think I don't know what you think, Singi. I think the issue is also sort of being able to do this quickly and in real time. I think that's still a little bit of the, maybe the bottleneck here, because
I think it's operational issues that we all have to figure out.
Yeah, because I think a lot of what we presented is cases from before, right? Historical cases. The number of existing and live cases that are being treated right now, maybe not in a huge proportion, although there are some, right? Yeah. Okay, great. Thank you. And then I think we've got a couple of questions from Professor Edison Liu. Your detailed historical data on individual patients can be used as virtual historical controls in your future clinical trials. Have you considered this to help streamline future trials of rare cancers? Okay, maybe Singi can address this first before I chime in.
So I think we do. I know a lot of people looking at data as controls for clinical trials. I don't think we really think about the trials downstream yet, but I think for sure we can look into this. Yeah. But I think Valerie can probably answer this question a little bit better.
Yeah, if I may chime in, I think that's also the other key use of putting together this data in this manner, because especially for rare cancers and for sarcomas, it is not possible to do a prospective, phase 2, phase 3 clinical trial with adequate trial recruitment over a very, very short period of time, short of having many centers involved. And of course, funding is a massive issue as well. So I think historically, the landmark trial in sarcoma, which was a phase 3 prospective trial, that simply asked the question of doxorubicin versus doxorubicin and ifosfamide, in sarcoma patients. They recruited only about 220 patients, and it took a good 11 years to recruit, despite 30 different centers being involved. Okay. And in fact, I would say the outcome of the trial was also not the most practice changing, although it is an important trial nonetheless, So I think the use of synthetic data in that sense and virtual historical controls are fantastic, because I think we're already designing trials in this manner for rare cancers moving forward. It's certainly something that we're discussing even within the Atlas Sarcoma and Rare Cancer Group. Professor Liu, I think you are on. Did you want to say something?
No, I just want to really, first of all, congratulate you. This is really a monumental task that was done extraordinarily well. But I totally agree with you. In the rare and orphan disease world, the use of synthetic controls is, you can't get away from it. And so, especially in the context of Asian-specific patients, you have your own reference. So I think that this is going to really accelerate your therapeutic potential going forward to do this kind of matching if you possibly can.
Yeah, absolutely. Thank you, Dr. Liu. Completely agree with you. And thanks for the comment as well from Professor Liu. By the way, this is a fantastic example of integrative systems medicine. Yeah, completely agree. There's one more question, if we may go to that. Oh, I guess this is a question for Dr. Wong. What is the current proportion of international patients receiving proton beam therapy in Singapore?
I don't have the exact data, but anecdotally, I would think that we do have international patients because we are a small city-state and yet we have free proton beam centers. But definitely, I think there's capacity for more, especially because we live in this region with NPCs, with HCCs, even lung cancers. So these are actually Asian tumors that can benefit, and not to mention the children with germ cell tumors, which is also an Asian tumor. So I would say that we still have capacity. And if we talk about dollars and cents, actually proton beam therapy in Singapore, because it's controlled by the government, is actually really affordable. For 30 fractions, about 60,000 Singapore dollars, is about 40 to 50,000 US dollars. And in the grand scheme of things, this is actually really affordable because if we look at biologics, this is really affordable. But as usual, because Singapore is an expensive city-state, so actually accommodation and logistics costs for these patients, especially with our strong Singapore dollar. So I don't really want to go too much into the economics of things, but yeah, we definitely have the capacity to treat more, and we should.
Okay, thank you, Dr. Wong. While we're waiting for any more questions to come through, I've got a question of my own, if I may. Okay, for Rusin, maybe firstly. So really, this is about precision radiotherapy for sarcomas and rare cancers, and I think you demonstrated multiple ways in which this is quite precise. I really was intrigued by the issue of re-irradiation, especially in the situation where you have a sarcoma that often develops right after radiation after a number of years. We always struggle in our tumor boards as well to think about whether or not we should give radiation again. And I think you mentioned quite interestingly that it could even be curative to re-irradiate someone who has a radiation-induced sarcoma. Is there data for that at all?
I'm sure there are case series, but this is a rarest of the rare, very, very often cancers. So I would think that the principles of curative treatment, if the patient is not metastatic, giving the patient the best option. So we want to reduce the treatment volume. So with protons, with carbon, we can reduce the normal tissues being irradiated. But I think overall, we have an informed clinical decision process with the patient. We tell the patient that, this is an option for cure, but we also have to emphasize that because this is re-irradiation, there are toxicities.
Yeah. Okay.
Yeah.
Yeah. Have you had any cases where you've actually managed to cure somebody with re-irradiation for a radiation-induced sarcoma?
Yeah, so like in my anecdotes, there were few, and in fact, I also wrote a paper with you.
Yes.
Yes.
Okay.
So in this really, really rare, often cancers, we should really try to get papers out, even though they may not be high impact because the numbers are small. Maybe Xingyi can help with some of this in the future.
Great. Sounds good. Okay. Thanks, Dr. Wong. I think, okay, it's more of a, I guess it's a question and a comment. Someone is asking whether we can get slides of the second presenter. I suppose we can discuss and see whether we are allowed to send it out, because I think some of it is also unpublished. But thank you for reaching out, Congo Baluti. Okay, I've got a question for Dr. Wu, if I may. So Singi, I think a lot of this is very translational. It has implications for future trials to be done, and maybe can even be used in an MDT. So there's already, quite a few clear, clinical translational examples. The work that we've done, has it also already been translated to patients? Or maybe I should ask.
Definitely like the published work that I've shown you earlier, whether it's a prognostic calculator that anyone in the world can use, as well as our drug screens that can point to certain drugs, and what are the mechanisms that we can actually translate that to patients. But really right now, I think the important part of sarcoma is really looking for newer therapeutics, because the standard of care, as you mentioned, is still doxo and so on. So it's, how are there more treatments available? I think there's a lot of biology that we can explore with our patient-derived cells. And we have various team members that look at, the immune environment on treatment. So these are really understudied areas, as well as resistance and so on. So we really work together on these various aspects of work. But for example, the bad eye inhibitors and all that is something that we really want to go forward, with Dr. Valerie and the whole team to, engage pharma on that as well, right? That is something we are continually learning to do. And the other aspect is really the MDM2 inhibitors on liposarcomas. So we know that the current trial didn't work out really well, but we are still, doing a lot of profiling to really understand, how MDM2, the kind of structural variations around that whole chromosome 12 region, what's really happening there. Is there a combination of genes and all that actually drive the proliferation of that? So I think from whether biological understanding to, all these specific questions that we drive with the whole team, our end goal is really translational. Yeah, but to work on tumor boards and all that with the dashboard, so we have experience on that. It's probably not too difficult. I think that lies in more of an operational workflow that we have to work with all our partners on.
Yeah.
Yeah.
Okay, thank you, Singi. May I also chime in and add actually the two publications that Singi highlighted. In fact, the precision medicine paper, that is already in use clinically. So in fact, this is available as a platform that can be used to inform treatments of sarcoma patients. And, the clinicians actually speak to us and discuss some cases where, we try to use functional precision medicine testing to inform what potential systemic treatments can work in these patients. So a lot of this is really about repurposing available medications that are already used in the context of other cancers. Simply asking the question of, if I add these drugs to these cancer cells from these patients, does it actually kill the cells or not? So that's actually been very interesting to see, because when we did that study, Singi, remember, we actually used this to inform treatment of every single patient on our study, from an n=1 basis, but also as a larger whole to accumulate these data to then come up with, what was the top potential hit, trying to look for common mechanisms of vulnerability across these cancers. So that's already one example of clinical application that's already come from this, right? And the other publication that we highlighted was really just using very well curated, as you rightly mentioned, clinical data, where you can now scan a QR code, right? And if you have a head and neck sarcoma patient sitting in front of you and you don't know what to say to the patient, you can at least go back to this pool of data and say, what can potentially happen, right, if you were to undergo radiation of a low dose or a high dose? What can potentially happen if you were to undergo systemic treatment, where, 5-year overall survivals, I think it was 7-year overall survivals, correct me if I'm wrong, could be tabulated and predicted based on, the cohort of patients that we had. And if you match those key variables, which were the ones that we identified to be significant, which affects prognostic outcomes. So I think already just pure clinical data alone can be quite powerful, and then layered on with, additional phenotypic, genomic, transcriptomic data, that becomes very, very powerful as well. Yeah. Okay. I think if there are no further questions, it leaves me to thank both speakers. Thank you very much for taking the time to give us these wonderful, wonderful talks. And also thank you to the whole Atlas group and Dr. Sakino for running the program, organizing the whole event. And may I also thank each and every attendee for taking, time out of your precious evening to come and join us on a very exciting series of talks. And feel free to reach out. I think we're all very friendly here, and happy to hear from you as well. If there's anything you want to ask, or if we could help each other, or if we could collaborate on something, happy to discuss this more as well. So thank you very much, everyone. And I hand the time back to Dr. Sakino. Yuta, please.
Okay, thank you very much. And thank you so much for speakers and facilitator Barry and Dr. Lucy and Dr. Sitting and every attendees. So see you on our next webinar. And thank you so much. And I may ask you for another round of applause to the speakers and facilitators, please. Thank you. Have a nice, good night.
Thank you, everybody.